Okay, so I want to spend a few minutes now talking about biological robots. We talked about rigid robots. We just talked about soft robotics. And along the way, the robotics community as a whole has been discovering that we can make robots with more and more different kinds of materials. In the 20th century, we typically used metals and ceramics and plastics and batteries and sensors and motors, but those rigid robots are limited. They can only change the way that they move. They can't change their own shape. We just talked about soft robots, which are able to change their own shape. And those soft robots are made from a wider range of materials. I just showed you a soft robot made from silicone rubber. Josie, who's here, might tell you a little bit about the work she's doing in the soft robotics space. Soft roboticists are able to make robots from more and more different materials. So a few years ago, my lab teamed up with Michael Levin and Doug Blackiston, two biologists at Tufts University. And Michael Levin's lab is very, very talented at building things from biological materials. So when we got together, we figured out a way to actually build robots from biological components rather than technological components. And that gave birth to the Xenobots, which I'm going to talk about now. You're looking at an AI-designed Xenobot or an AI-designed biological robot on the right side of the screen here. We published our first paper about Xenobots in January 2020. And as you can imagine, if you make very, very small robots out of biological materials, we got a lot of interest from the global press at that time, a lot of coverage about our work on Xenobots until 2 months later, until March 2020, and another very small creature showed up that stole the limelight away from our Xenobots, and people focused for the next 2 or 3 years on another very small creature. Anyways, let's talk about Xenobots today. Just to give you a sense for what you're looking at, Xenobots are very, very small. They're about 1 millimeter across. So you can see Sam here holding up a biological slide, and captured underneath that glass right here is a Xenobot. Hopefully most of you can see this on your screen. These are tiny millimeter-sized robots. What are they made from? I mentioned they're made from biological materials. What biological materials are they made from? They're made from frog cells. And Xenobots are made from a particular type of frog. It's called, in Latin, Xenopus laevis. So we took the xeno from Xenopus, and we put xeno together with robot or bot to get Xenobot. In this very first experiment, our biology colleague, Doug Blackiston, told us that he was able to extract from very early frog embryo, basically a frog egg. He was able to extract 2 cell types, frog skin cells and frog heart cells. Frog heart cells, like human heart cells, are cells that when you put together, they contract and expand in volume, not unlike the soft robot that I just showed you. The red patches in the soft robot I just showed you, very much like heart tissue or muscle tissue that expands or contracts. And that muscle can be used to obviously deform the body of an organism, or in the case of the heart, all of those heart muscle cells get together and contract together to pump blood out of the heart, or all of those heart cells expand together to pull blood into the heart. So what you're going to see in a moment is the AI learning how to use the frog heart cells as motors. The AI is going to figure out how to combine these little pieces that expand and contract in volume together with these other pieces, which are frog skin cells. These pieces are soft and passive. They get pulled and pushed by the soft, active heart muscle cells. Okay, so here we go. Our biologist colleague told us, or told the AI actually, that there are 2 building blocks. The AI can put together these 2 types of cells in different combinations to make different shapes. So we're looking now at the AI, and we're back to an evolutionary algorithm. So this evolutionary algorithm is running on our supercomputer. And at the beginning of this experiment, the AI is putting together simulated heart skin cells, the blue boxes, together with frog heart cells, which are the red-green voxels or cubes. You can see three of the evolutionary algorithm's initial random guesses. This first guess in the upper right, the AI has just by chance put mostly frog skin cells together, the blue stuff, and you can see that the blue stuff is being pulled and pushed by the red-green cubes. When the cubes are red, those cubes are getting a little smaller, and when the cubes are green, they're getting a little bit bigger. You can see those red-green cubes pulling and pushing on the blue cubes. The task that we've asked the evolutionary algorithm to solve, as always, is figure out how to put these two types of building blocks together to make a robot that moves from the left to the right as quickly as possible. You can see that this particular Xenobot in the upper right is not doing a very good job at moving from the left to the right. Its second initial random guess, it comes up with another random guess. In this case, that Xenobot is just a big ball of muscle, of heart muscle cells. Despite the fact that it can move a lot, it doesn't move to the right at all also. The third one in the bottom right, although it's really hard to see in this video, you'll have to trust me, this particular bot moves 1/10 of a millimeter to the right. So even though it's a random guess, this third random guess does a little bit better than the other two guesses. And this is what an evolutionary algorithm needs in order to work well. One individual in the population, in this case, we're just looking at a population of 3, that one individual does a little bit better than the others. So what the evolutionary algorithm does at this point inside the supercomputer is delete the first and the second random guess. The third random guess has survived. The evolutionary algorithm takes that third random guess, that robot in the bottom right, it makes a copy of it. And when it makes a copy of that surviving robot, it introduces some small random mistakes. It adds or removes a blue cube, or it adds or removes a couple of red-green cubes. So now we have a new child robot that looks very similar to the robot in the bottom right, but it's also a little bit different. Not unlike actual children and parents, the child resembles its parents, but it's not identical. That same robot in the bottom right makes a second child to replace the robot in the upper right. It's producing 2 children. So now in the population, we have 3 robots. We have the parent in the bottom right, and we have 2 children in the upper right and middle right that look and act similarly to the parent. And because those children suffered a small random change, most of the time those children move more slowly than the parent. Those children are also deleted, and the parent is given an opportunity.
We're going to Google Xenobots, and we're going to click on images. Meet the Xenobots, virtual creatures brought to life. Computer scientists and biologists have teamed up to make a new class of living robotics that challenge the boundary between digital and biological. This right here, this is a Xenobot. Derived from the skin cells of frog embryos at an early stage of development in the laboratory. If the last few decades of progress in artificial intelligence and in molecular biology hooked up, their love child, a class of life unlike anything that has ever lived, might resemble the dark specks doing lazy laps around a petri dish in a laboratory at Tufts University. Douglas Blackiston, a biologist, pointed to one just a little wider than a human hair. Squint, and you could just tell it was moving. But, under a microscope, the blob was racing up and to the left. "He's a lighter," Dr. Blackiston said, then caught himself. "It's a lighter color." Strictly speaking, these life forms do not have sex organs, or stomachs, brains, or nervous systems. The one under the microscope consisted of about 2,000 living skin cells taken from a frog embryo. Bigger specimens, albeit still smaller than a millimeter-wide poppy seed, have skin cells and heart muscles that will begin pulsating by the end of the day. These are all programmable organisms called Xenobots, the creation of which was revealed in a scientific paper in January. They are named for the African clawed frog, Xenopus laevis, which supplies all their cells, and the suggestion encapsulated in the prefix that something strange, alien, is at work. A xenobot lives for only about a week, feeding on small pellets of yolk that fill each of its cells and would normally fuel embryonic development. Because its building blocks are living cells, the entity can heal from injury even after being torn almost in half. But what it does during its short life is decreed not by the ineffable frogginess attached to its DNA, which has not been genetically modified, but by its physical shape. And xenobots come in many shapes, all designed by robotics and computer simulations, using physics engines similar to those in video games like Fortnite and Minecraft. Xenobots with a fork or snowplow-like appendage in the front can sweep up those loose particles in a petri dish overnight, depositing them in a pile. Some use legs of a sort to shuffle around on the floor of the dish. Others swim, using beating cilia or link up blobby appendages and circle each other a few times before heading off in separate directions. All of which make xenobots amazing and maybe slightly unsettling. Golems dreamed in silicon and then written into flesh. The implications of their existence could spill from artificial intelligence research to fundamental questions in biology and ethics. We are witnessing almost the birth of a new discipline of synthetic organisms, said Hod Lipson, a roboticist at the Columbia University, who was not part of the research team. "I don't know if that's robotics, or zoology, or something else." Now let's go back to Google Images. We're going to click on this right here. And we're going to read. Meet the Xenobots, robots that can reproduce. Life has to reproduce in order to survive. Organisms have developed a variety of methods for self-replication over billions of years, ranging from sexual mammals to invading viruses to budding plants. The first ever self-replicating living robots have been made possible by scientists at the University of Vermont, Tufts University, and Harvard's University Wise Institute for Biologically Inspired Engineering, who has discovered a completely new method of biological reproduction. These computer-designed and manually assembled organisms can swim out into their tiny dish, find single cells, gather hundreds of them together, and assemble quote-unquote baby xenobots inside their Pac-Man-shaped mouth. These xenobots, a few days later, grow into new xenobots that look and move just like themselves, according to the same team that built the first living. These new xenobots can then venture out, locate cells, and create duplicates of themselves. Once more, and once more. Come, Let's learn how Xenobots reshape our understanding of genetics. What are Xenobots used for? Diving into the strange world of Xenobots, we know that Xenobots can be programmed, and therefore, a plethora of opportunities are forthcoming. Because Xenobots are made of stem cells, their capabilities are determined by their design, which makes them extremely versatile for a variety of uses. As such, Xenobots are redefining biotechnology. Healthcare. Even though Xenobots currently have simple behaviors, they are still developing. In the future, it is hoped that they will be incorporated into the human body for regenerative or targeted treatment. Furthermore, unlike typical metal or synthetic medical equipment, they are composed of living tissues that will eventually disintegrate. They are especially safe for humans to use because of this. They may also be utilized to improve internal drug delivery because they may endure for weeks in watery settings. Cleaning up our environment. Researchers came to the conclusion that biological robots could ultimately be taught to target microplastics and help with the cleanup of the 50 to 75 trillion particles of plastic that are fouling the ocean, inspired by the Xenobots' collective corralling abilities. The same technique can be used to detect and get rid of radioactive substances found in the environment, such as nuclear waste, or it can be used to monitor and maintain ecosystems using Xenobots. The ability of Xenobots to adapt to organic settings without contaminating them is crucial. Producing more Xenobots. These living robots can create new Xenobots by clumping together loose cells, as scientists have discovered. This is how Xenobots reshape our understanding of genetics. Biological robots with autonomy. It is hoped that this study will progress to the point where Xenobots can function on their own without assistance from humans. They do, however, come with hazards and ethical dilemmas that should be taken into account, just like any biological innovation. How do Xenobots reproduce? Xenobots are the first robots made from living cells. Not only can Xenobots carry out basic duties, but they can also duplicate themselves. In a follow-up study released in 2020, the same research team revealed that the living robots would cooperate to create baby Xenobots by spontaneously gathering hundreds of loose, single cells thrown into the petri dish, which was first provided as a feedstock. Diving into the strange world of Xenobots, parent Xenobots use a corralling motion to push hundreds of individual stem cells into a pile while swimming about on hair-like cilia. The trove of cells is compacted and formed into progeny over a period of roughly 5 days, after which they are expelled from the parents' wedge-shaped mouths. The following generation will change into a fully functional Xenobot, which will have the ability to make more copies, and so on. This kind of reproduction, known as kinematic self-replication, has never been seen before, according to researchers. In fact, it wasn't even considered feasible because this process has only been observed at the molecular level and only in multicellular animals. This is also one of the ways Xenobots are redefining biotechnology. How can Xenobots be programmed? Xenobots are the first robots made from living cells. The first step in the entire procedure is to separate the embryonic cells from the frog's egg. Two of the many intriguing characteristics of the cells are pertinent to our work. First of all, these cells adhere to one another. They will come together into a ball if you leave a cluster of them on a culture plate. They will even adhere to one another and create a ball of cells if you completely separate them and leave a collection of them in culture. And it is sufficient to initiate motion through the entire ball of cells. For ease of reference, we will refer to these as mobile balls of cells, or MBCs. An MBC can travel in a circle in a cultured dish for up to 2 weeks without running out of energy if there are no obstructions. The living robots are these. The novel aspect is that a group of separated cells was added to the plate containing the MBCs by the researchers. Additionally, as the MBCs revolved, they formed a cell cluster at the center of the circle they traced. Imagine the mobile ball of cells as a donut, with the cells forming a pile in the hole. Additionally, a new cluster would start to form from these free cells because they naturally self-adhere. The new clusters were too small to be of any use on their own. However, they might be taken off of this plate and put on a fresh one that has a lot of extra cells. By absorbing some of these extra cells, the little clusters would expand and eventually get to the point where they could move as well. How long do Xenobots live? A Xenobot, which consumes the tiny yolk pellets that fill each of its cells and would typically support embryonic development, barely has a week or so to live. Even after being ripped nearly in half, the creature may recover from damage since live cells make up its structure. The ramifications of swift technical advancement and intricate biological engineering are a source of concern for many. In order for humanity to endure into the future, we must get a deeper comprehension of how intricate characteristics somehow arise from basic principles. A large portion of science is concerned with regulating fundamental laws. Furthermore, we must comprehend the high-level regulations. Future societies will therefore need to develop a deeper comprehension of systems with extremely complex outcomes.
So when you can carve out the human with synthetic biology over time by resurfacing their cellular structure over time, with more and more layered synthetic wireless tissue engineering, synthetic biology, tissue scaffolding, then eventually you're going to be able to turn the skin into metal, plastic, or whatever they want because the skin no longer holds its electrohomeostasis.